Okay, so now we've collected saliva sample on this swab, completely absorbs it, there's no dripping or anything. It's important again to emphasise this, it isn't a quick swab around a dry mouth like you see when they're doing DNA testing for forensics. This swab has to be completely saturated in saliva, and when that happens it's completely squishy, as you can see, and soft. It takes about 25 to 30 seconds. And when that's occurred, you can flip the lid on the saliva collector, and by pushing down into the base you will discharge the saliva into the collector at the base. Now I'm just going to clip that closed like that, and if I can show you now how much saliva we've collected in there. It's not showing particularly well on camera, but you can see it's approximately half filled, and you could expect to get at least that amount if possible on each collection to be able to run the two tests. And to do that, just unscrew the lid, and this is basically a squeezable pipette now. Invert it, add 3 drops of your solution to each of the sample wells. Seal that. Now you've got the ability to put date, initials and ID on here. As we showed you earlier in the packs, you also have some seals which you can sign across and seal across the opening of this saliva sample, so you can actually identify that and store it for confirmation testing should that be required. Now what you'll see happening here on this test membrane, if I bring it closer to the camera, you'll see that pink dye travelling upwards towards the C line indication and the top of those test windows. When you add the saliva, you start your timer, and indicated in the instructions is a correct read time, and you need to read the test lines showing at the correct read time for the individual test, and it does vary between different brands and different saliva drug tests, so you need to know the correct time for interpretation of the results. Now you'll see on this we've got some test lines forming already. A negative result on saliva drug screening is indicated by test lines present in the presence of a control line at the top of that membrane. You've got to remember this is negative binding technology, so if there is any drug residues or metabolites in the saliva sample to indicate blood consumption, they completely bind the membranes at those test line areas and do not allow any dye to form at those areas. So a positive result is indicated by a control line with the absence of any test lines showing at the indicative read time. So I'm just going to stop this video now, wait for the determined read time to occur, and show you the results showing at the correct read time for this test membrane.